In this video, we'll see very briefly about retroviral transduction, about macroinjection, and about the liposome mediated gene transfer. Retroviruses are RNA containing viruses. These viruses could be used for transduction. There is a very high efficiency gene transfer. They infect the host cells spontaneously and the transfer is permanent, and they are inserted by normal cellular processes and does not harm the host or does not cause any genetic alteration in the host. Macroinjection is done using injection needles with diameter far wider than the cell diameters for delivering the DNA into the wound sites within the tissues. It is developed for cereals which are unable to regenerate into the whole plants. 0.3 ml DNA solution is injected above the tiller node region 14 days before meiosis. Advantages: it does not require protoplast. It is a simple and cheap method. Gene transfer is done into cereals that do not regenerate easily. Disadvantages: it is less specific, less efficient, and low frequency of transformation. In gene transfer using liposomes, liposomes are phospholipid bilayers which are concentric in nature and they are microscopically small sized so that large number of plasmids can be enclosed within the liposomes. Using polyethylene glycol, liposomes with recombinant DNA are fused to the protoplasm and the mode of transfer is endocytosis. Studies have been done with tobacco, petunia, carrot, etc. The method involves the following steps: adhesion of the liposomes to the protoplasm surface, fusion of liposomes at the site of adhesion, and release of plasmids inside the cell. The picture of the liposomes are shown. In case of multilamellar liposomes, the outer membrane will fuse with the plasma membrane and the inner membrane could be impregnated with nuclear proteins so that the inner membrane will fuse with the nuclear membrane, delivering the recombinant DNA into the nucleus. Advantages of this method are simple method, low toxicity, long stability, large DNA can be delivered, long stable storage of DNA fragments in the liposomes, protection of DNA, RNA from the nucleases that lyse them, and liposomes never disrupt the integrity of the cell. It is applicable to various cell types. They never interfere with the immune system. There is a high level of reproducibility, and liposomes with desired property can be made by impregnating with specific proteins. Preparation of liposomes, water and oil type emulsion. Brief sonication is done with phospholipid and aqueous buffer in a large volume of organic solvent. For oil and water type emulsion, the phases are reversed. And finally, evaporate the organic solvent to get the liposomes. Monoclonal antibodies could be used to direct the DNA to the target cells. Monoclonal antibodies recognize the surface antigen of the target cells and specifically binds to it along with the liposome. In ultrasonication, sound waves are used. Cultured explants are sonicated with plasmid DNA containing marker genes, and the regenerated callus is grown in a selective medium to generate transgenic plants. This has been done in wheat, tobacco, and sugar beet.